Hello, everybody. My name is Tony Heil with ALS United Mid-Atlantic, and this is our Goals podcast, where we talk about the very different perspectives on ALS, from research to caregiving to living with ALS, so you can understand the whole wide spectrum of how we are tackling this disease to help people living with ALS and their families. Today, I am really excited to be talking to my friend Carol Hamilton from ALS TDI, the Therapy Development Institute. Carol spoke at our 2024 Help and Hope event, and she is going to be speaking at our 2025 Help and Hope event, though you may be listening to this after that too, so it's always great information coming from Carol. She explains things about research in a really understandable way, and she's not going to go into the details of how to splice RNA, but we're going to be talking about really important things about research, like biomarkers and risk factors, and how we can work together as an ALS community. ALS TDI is based in Massachusetts, and Carol started as a volunteer. She's helped secure funding for research, and she has done a lot of work in her own professional life. She is a vice president of development, and she studied at Oxford University and graduated from the Georgetown University School of Business. She also has started her own company before, so she has a lot of great experience that is going to help lead the ALS cause for all of us. So, Carol, thank you so much for speaking today.
I'm so happy to be here, Tony.
Well, terrific. So, as I said, you started as a volunteer. You have your own ALS connection, and I would rather you share that story than me tell it for you. So, what brought you to the ALS cause?
Let me start with this. It's kind of crazy because when I was a kid, I had two different reoccurring kind of scary dreams, and one of them was being stuck in a box and not being able to tell my parents I was in there. And when I was given the opportunity to help in the ALS space, I kind of felt like I was eradicating my childhood nightmares. It was kind of meant to be. I had, unfortunately, a dear friend whose younger brother, who I knew, young man in his 30s, two little kids, diagnosed with ALS, and a friend of mine said, "People are dying, we need your help." And I went in and I met the founder of the ALS Therapy Development Institute, we can call it TDI for short, Jamie Haywood, whose younger brother Stephen had been diagnosed with ALS at 29, and they started ALS TDI in their family basement, and now it is, of course, the largest independent ALS research institute in the world. So I came in, met him. He said, "Do you want to see a world without ALS?" And I said, "Yes, let me try to help." So I had worked in government affairs in college, and that was kind of the first hat that I wore here, helping to establish funding at the Department of Defense, the ALS research program. And since then, I've worn a whole bunch of different hats, and I will wear whichever one will be able to help this community the best and the fastest for the rest of my life. That's it.
If you don't mind, you mentioned about the ALS research project at the Department of Defense that's done some great research, and there's other research being done through the Department of Defense because a lot of diseases are military connected. ALS is a service connected disease. How important is that project, the CDMRP as we call it?
It's incredibly important, and the genesis for that program, as I said, was some work that we were doing with, at the time, the comptroller at the Department of Defense, and he aggregated some funding to first put that program together at CDMRP, Congressionally Directed Medical Programs, and this was after a lot of work helping Congress to understand the military correlation of ALS, because at the time, the program was started with the breast cancer lobby, which was fabulous, but ALS was, as you said, a service-connected disease, so we thought it was really important for DoD and Congress to understand that. So we first got funding at about $2.5 million, and over the course of time, that funding has grown. Last year, $40 million. The continuing resolution for 2025 did include money for CDMRP. We are remaining hopeful that we will get the $40 million for 2025. ALS TDI has received funding through that program, as have many other great research organizations and ideas that are leading to therapeutics moving forward in the clinical space. So it is paramount to our ability as a research community to move forward at the speed that we've been at for the last couple of years.
And that reminds me, it seems like now more than ever, the ALS groups, whether it is nonprofit organizations or research centers, universities, and drug companies, it feels like there's more collaboration than ever before, more communication between people. We've had you speak at our events, some of our doctors talking together and connecting more than before. Do you sense that too, that there's more unity in terms of collaborating now?
Oh, Tony, absolutely. And a big part of it is the perspective and approach of ALS United's. It's really been amazing for us and other organizations to be able to work with you guys who are on the ground. We certainly see it in the research space. It's a building block of who ALS TDI is. And we can talk later about some of the different ways that we collaborate. But you're absolutely right that the hopefully positive effect of moving forward with the ALS research program at DoD is a result of all of our organizations getting together and advocating. It's a much better tenor right now in the community, and I'm delighted by it.
And one of the ways that we work together is by connecting people with ALS to participate in research, to know about clinical trials, maybe to know about things that you're working on. And so research is very empowering to those who are living with ALS. You've talked with people like our friend John Russo, who's living with ALS and very active in both of our organizations. How is research and those clinical trials, how are those things empowering to those living with the disease?
Oh, I think it's incredible. I mean, first of all, our ALS community truly does seem to have a magnanimous nature that I don't know exists outside of it. I've never seen anything like it. There are people facing this horrible disease who say, even if what I do can't help me, I want it to help other people. That is incredible. And it is really the only way that we can move research forward is by learning from people with ALS. I think it's particularly empowering when you have a disease that can be isolating. It's hard to get around, it's hard to leave your house. But if you can participate in research digitally, online, or through a virtual trial, then you are having an effect on the space. I think it's really liberating and empowering for everybody to engage. And there are all different ways that people can participate in research. We want to make sure that while people are doing it, that we decrease the burden on them as much as possible while getting as much information as we can. It's important, as I said, for all of us on the research side to learn from people with ALS. They have the answers in their bodies, right? But as I think I joked at Help and Hope last year, we don't want to cut them open to get that information out, right? That's no good. So we've got to figure out ways to extract all of the learnings that are inside all of the people living with ALS and apply them to research to develop treatments faster. And there is absolutely the opportunity for every single person with ALS to be able to do that and have that impact.
Well, that's a really good segue to an important topic you've discussed, and that's biomarkers, because you said you have the answers inside of you. And I don't think people understand what a biomarker is. Just if you were talking to a regular person like, I guess, me, how would you define a biomarker?
Well, there are a couple of different types, right? But let me use some examples that might be familiar to people. Insulin levels are a biomarker in diabetes, right? Sugar levels. Tumor size is a biomarker of potential drug response in cancer. So there are actually 7 different types of biomarkers. Let me whittle it down to 3 just for conversation, you know, we can talk about diagnostic biomarkers, right? Helping to diagnose a disease. We can talk about prognostic biomarkers that tell us, is this person perhaps going to be a fast progressor or a slow progressor with ALS? And we can talk about a third type, like I mentioned a minute ago, is a biomarker of drug response. Is a drug that we are giving a patient having an effect on that patient's disease? So there are ways of knowing how the disease is moving forward in each person and how the treatments that we are giving them are affecting their disease.
So there's blood-based biomarkers. Is that like measuring things in the blood, or exactly what would that entail in terms of measuring the prognosis and the progression of this disease?
Yeah, there are blood-based biomarkers, and ALS TDI that's a big, one of the big things that we do is we, through our ALS Research Collaborative program that, you know, many people in your group and constituency participate in, we're able to send phlebotomists to people's homes once a quarter and get blood. Comes back to ALS TDI, to our 36 scientists, who then analyze it and look at different protein levels. You know, one biomarker that is talked about a lot in the ALS community is neurofilament light chain, NFL for short. And that is both a serum and blood-based biomarker, excuse me, that you want to be able to measure over the course of time. So people being able to give blood temporally over the course of their disease for us to be able to follow their NFL levels is incredibly helpful for so many ways. First, imagine, Tony, if one day we were able to validate NFL as a biomarker. And for those who may not be seeing us, I'm using air quotes when I say validate. That is a term that has a formal definition with the FDA. And that would mean, imagine if in a trial we were able to say, okay, your NFL levels, Joe, who is in this trial, have risen or decreased, and that is telling us that this drug is having an effect. Now, we need to validate it with the FDA so that they can accept neurofilament light chain, NFL, and digital biomarkers that I'll talk about in a second as an endpoint in a clinical trial. We saw Biogen use this with their Atlas trial for tofersen. Now, here's a really cool thing. Neurofilament in SOD1 patients in particular, one mutation, probably the mutation that we know the most about because it was discovered the earliest. In people with SOD mutations, their NFL levels can change before they start to see any physical symptoms. So what Biogen did with this, which was awesome, is they began following asymptomatic, pre-symptomatic genetic carriers of SOD1, and they would they would take their blood, and as soon as they saw their NFL get to a certain level, they would begin treating them with the drug that they have, Qalsody, that is an FDA-approved treatment for people with SOD1 ALS. Now, I have friends who have gone through this, who are SOD1 patients. They had no idea that their disease had begun to rear its head. They felt no physical difference whatsoever. But their blood told them that their NFL was starting to rise. So NFL, by the way, is what happens when a motor neuron dies and it starts to kind of shed itself into the blood. That's what the neurofilament light chain is measuring. So people are able to get treated earlier in this Biogen example. So imagine that, right? Imagine we can begin to understand risk factors for people with ALS, like a genetic background, and treat them earlier. That's going to matter. So, yeah, blood-based biomarkers are a big deal.
It reminds me of being a parent and you know your kid pretty well. Like if you have a toddler and you know, oh, when they start getting grumpy, that means that they might have a headache. So they don't have a fever yet, but we're going to check on this because I know these kind of signs in my child. So you kind of find these signs for a disease like ALS, like you were saying, and now you can kind of look and say, we know this kind of family history, we know this other item in them, and now we can kind of check that out because we understand people better from all of the data we have.
Exactly. And in your example, what that would mean too is you have that kid's Tylenol ready to go, right? And you are going to save them some misery by being ready with that drug. That's exactly what we're talking about. Yeah, and there are, we can talk about risk factors a little bit too here because obviously a genetic mutation that you have carry in your family in familial ALS is a risk factor. ALS TDI is collaborating with Pat Dolan, who is a person living with ALS, who's a geospatial analysis expert. How about that? Think MAPS. Through a grant from the CDC, ALS TDI, Pat Dolan, Dr. Danielle Boyce, we are looking at lifestyle risks in ALS right now. Being able to use that data that you referenced from our ALS Research Collaborative, where we are doing exactly what I talked about, following people with ALS longitudinally and finding ways to extract that information that they have in a low burden way. So we are able to, hopefully, at the end of all of the study, identify a few more risk factors for ALS and then think about how that matters and how we can, by knowing that, get to treatment earlier or maybe begin to subset patient populations that will be responsive to one particular drug or another. I mean, that's a big part of it, Tony, right? This is a very heterogeneity. There's a lot of heterogeneity in this disease. We need to subset people so that this group may be responsive to this drug, as we see in SOD1 and Qalsody. This group may be responsive to this one. That's a big part of the work that we're doing with the help of the ALS patient population. They're giving us the information to build that data set.
And the broader scientific community then might see what you are doing or what other scientists are doing to find biomarkers for ALS. And it sounds like it takes a lot of creativity to be a scientist in any of these fields. So you see, Carol's group and the doctors at ALS TDI, they looked at this kind of blood level or iron level or whatever. Maybe we should look at that kind of idea for this disease or for Huntington's or for something else. And it may be a very different drug, but it kind of gives them a map for something to look at for other kind of science as well.
You're absolutely right. As a matter of fact, yesterday in the lab, I had a gentleman from a pharmaceutical company that's developing a therapeutic for ALS on the clinical level. And that company has a program in epilepsy as well. And, completely different population, right? A gazillion times bigger than ours. But again, heterogeneity. So he was learning about the ALS Research Collaborative program that we have at ALS TDI, where there's 45 terabytes of data, all given by people with ALS. And he said, this is the dream of the epilepsy population. They don't have this. We had kind of the prescient vision to start this program kind of not flying by the seat of our pants, right? Not knowing what we didn't know, but we knew we had to start. So it was a proud moment yesterday, as he said, would I be able to connect some of the people in the epilepsy research field to you guys so that you can help them build this type of program there. So yeah, there's collaboration across disease indications, and there's the opportunity now to take data like we have that people with ALS have provided for us and share it with every researcher across the globe. That way, like you said, if somebody else is looking at something a little bit different than we are, they can go into that data set and they can mine it and they can accelerate their own work because we're going to give them that. We want everybody to move faster. And I think we're finally at a place where we have the technology and the will to do that.
It feels like a video game from where you don't know how to find the big bad guy or find the things, and so you look up the guides. Maybe you go online and like, oh, this is what I need to do. There's a Konami code, as people would say. One of those codes, like you said, are digital biomarkers, because I want to go back to that. It's so important. I heard from some of our other researchers about some really neat ways to measure progress in ALS, from handwriting samples to walking and like measuring your walking gait, and of course a voice progression and the changes in your voice.
Yep.
How are you using digital biomarkers to understand ALS?
Great question, and I'm going to use your kind of prelude into this and combine it in my answer. So when we were starting the ALS Research Collaborative, we had an idea that vocal progression in ALS dysarthria could be important, right? So what do you do with it? You and I, all of us, can hear the difference between someone who was diagnosed yesterday or someone who has had ALS for 12 years. We can hear it in their voice. But how do you quantify it? And does it matter? That's a qualitative assessment. So we knew we wanted to follow people with ALS and their voices. We began by having everybody as part of this program basically call into an 800 number. I'm probably dating myself by putting it that way, but all the same, and repeat certain phrases. And we would have them do that over the course of time, longitudinally. And of course, their ability to repeat those phrases is going to diminish because of the dysarthria caused by this disease. So what do we do with that then? We used to listen to those recordings and we would each kind of guess how far along somebody was. Well, that's very unscientific, but we still knew it was important. So we worked with Google, actually, right? They're the kings and queens of vocal AI, understanding, voice in the digital era. And they used machine learning to develop algorithms to quantify vocal progression over the course of ALS. It's really quite amazing. So really, they'll take the sound waves and they'll treat dysarthria and ALS almost like an accent, a French accent, a German accent. Then they look at the pictures of how people are talking and they can quantify that, turn it into an algorithm, and rate something over the course of time. One of the things that we have found, so there's an example of really not knowing what we were going to do, but knowing it was probably important and saying no one else is doing it. Let's not duplicate efforts. Let's take a shot. Let's try. In this case, it was a really good bet. So now, with those algorithms, with the help of machine learning and Google, we are able to rate vocal progression, and we are finding that it mimics the ALS FRS score, right? In an objective, scientific way. We all have talked about issues that we have with ALS FRS. It can be subjective. Different people administer it different ways. It puts equal value on things that, are not always equal. Breathing might be more important than handwriting. So everybody's always looking for a more objective measure of ALS disease progression, a biomarker. So in this way, this is raw data coming from people with ALS that we are able to develop algorithms for that is showing us that while we know the ALS FRS isn't perfect, it's what we've got, and these ratings are following the ALS FRS. So imagine if one day, Tony, a person can enter an ALS trial and determine if a drug is working for them by calling an 800 number and saying, I owe you a yo-yo today. It could one day be as easy as that. So again, that's a way of using a digital biomarker, decreasing the burden on the ALS population to be able to contribute their data and participate in research, it's really quite astounding. We're also able to do it. Another potential digital biomarker is using what we call accelerometry. So that's wearing, wireless devices on wrists and ankles, sorry. They look like clunky Fitbits. You wear them for a week, a month. And we are able to wirelessly collect movement data from people with ALS, again, over the course of time. Now that we've been able to rate too. MGH just published on the data that we've collected using these accelerometers. Again, it could be a clinical endpoint one day in a trial, where if you participate in trial X, you wear these accelerometers, these clunky looking Fitbits, for however long. Sure, you take them off to shower. Sure, you take them off for your son's wedding. However, they're a great conversation piece, so wear them if you dare. But low burden way to collect data that could then be validated by the FDA to use in trials as a digital biomarker. Again, you go into trial Y, you wear these things, it gets graphed, and we determine if that drug is working for you. It allows us to make trials less expensive. It allows us to decrease the burden on patients, and it allows us to make trials shorter and more specific. So digital biomarkers are a big deal coming down the pike in the ALS research world.
That's really exciting, and it means that more people can participate. It's really reducing those burdens, and I think that'll, like you said, it'll help us speed research. To that, you mentioned in another conversation about the different models of ALS that you may look at. You're looking at different descriptions of them. What does that mean, and how are they used in your research with different models of ALS?
Great question. I remember when I came into the world of ALS in 2005, the SOD1 genetic mutation had just been discovered by Bob Brown, then at Mass General, and turned into an animal model. Without models of ALS, we can't know how drugs are going to work before we put them in people, right? And drugs are dangerous, so we don't want to risk people by not doing the preclinical work, which is a lot of what TDI does. So I'm going to define that for you. Everybody hears the terms clinical trial. That means human, right? Preclinical kind of means pre-human. And that's all of the work to determine the efficacy and safety of a drug in models of ALS before we put them into people, so that we know, or we can be assured as possible, that a drug is not going to hurt somebody. So Bob Brown discovered the SOD1 mutation as relevant to ALS. They built a mouse pretty quickly. And the way that you build an animal model, and in this case the SOD1, it was the first animal model of ALS, is you have to genetically modify the animal so that it has a progression of disease that is similar to human disease. So, for example, in a SOD1 mouse model, there we have, and others do too, a colony of animals. We get 100 mice in every Monday. I'm being somewhat cavalier with my numbers, but it's close enough. And in that colony, their parents have been given copies of the SOD1 mutation, about 27 copies, to allow the offspring to exhibit ALS as we know it in humans, because mice don't naturally get ALS, so we kind of have to give it to them. And I know that sounds rough for animal lovers, of which I am one, but I'm an even bigger human lover, and these animals are bred for this purpose. And I will tell you too that in our hands they are incredibly well cared for and appreciated. They are key to the future of having treatments for ALS. So there are different kinds of animal models. There are a number of different mouse models at this point in time, SOD1 being the one that we know the most about. That is the best characterized, as we say, the best understood. We can anticipate it's how the animal progresses because it's been around the longest. There are other mouse models of other various mutations that are relevant to ALS. Profilin 1 is one, for example. There are other types of animals that are used as models. Zebrafish, C9orf72 is the largest piece of the genetic pie in ALS, and we have C9orf72 zebrafish. Eventually, we'd like to have a C9orf72 mouse, but so far, as groups work to build a mouse model with a C9 mutation, so far it is not capitulating disease in the way that humans do. So the field is still working on that, but we do have the fish model. Then there are cellular models of ALS, right? So if you think about it, if you're testing a drug, which is a very expensive process and takes a long time, you want to start in cells. They're the cheapest. There's the most of them. And you determine, okay, did it pass that bar? Then you might want to use them in fish, right? They're cheaper, it's faster, they're smaller, you can fit a lot of them in a lab if it works there. Then you're going to go into mouse models, mammal models, right? Those are a little bit more expensive. You know, a preclinical experiment testing a drug in a mouse model will cost over $250,000. So if you're going to get a no, you want to get it earlier in that funnel, right? Back in the cells. Now you still may get it in the mouse, but first you want to try it in the cheapest, fastest, next cheapest, fastest, and then the big way. And you really do have to test drugs in animal models because they have systems like humans do, right? Cellular models are phenomenal because you can test a lot of drugs at the same time. But cell models don't have pulmonary systems. They do have circulatory systems, but they don't have all of the processes, the inflammation processes that we have going on. So eventually you do need to get it into an animal model. So if we have a drug that passed cellular model testing, fish model testing, and it's going into, let's say, a SOD1 animal model, we are going to design that experiment with probably about 40 animals over the course of time. It will take about 100 days to know if something is, showing any sign of efficacy or not. And then you have to really do the work after the experiment to look at all of the tissues and see the effects, right? You do tissue harvesting, you get it under a microscope. Did the drug get to where we wanted it to go? Even if it didn't have an effect, even if it didn't extend survival in an animal, you want to look at that because perhaps you need to rerun the experiment. So that's kind of the process of it. And then if a drug works, let's say, in a mouse model, we're going to do it again, and we're going to do it again, and we're going to do it again, because the last thing we want to do is send something forward that could potentially hurt a person with ALS. They're my favorite kind of people. And then, from there, you go to the FDA, and you say, okay, FDA, here's all of our data. We ran this in cellular models from people with ALS. We ran this in fish. We ran it multiple times in mice. We think that this warrants moving forward into a clinical trial. What do you think? Again, of course, it's far more formal than that. They then give you a set of experiments that they want to see you do. It's called IND enabling work, investigational new drug enabling work. Before you can run it in humans. So that's all of the work that's happening before something even gets to clinical trial. It's where most drugs fail. We see an awful lot of failures in clinical trials in ALS, but a heck of a lot more of failing before you even see them at all.
So with the mice, when they do the voice progression test, do you dial the phone for them to call in, or do they use their little tiny feet and hands?
These are really talented mice. They're really talented. As long as the disease hasn't progressed, they can hit a phone no problem. You should hear those squeaks.
That's amazing. Well, I think it's really fast and shows the complexity of ALS research, because people think you just come up with a drug, you put something together, you give it to a mouse, and then a few weeks later you know something. And obviously it takes years and a lot of money and resources to make that happen.
It does. The average, they say, in the drug industry is about 13 years and $2 billion for a drug. Now, that's both clinical development and preclinical development, right? And especially in disease communities where it's a very large population, those trials are far larger than ones that we see in ALS. It gets, even more expensive. So while, I certainly from time to time demonize the pharma industry like everybody else does, there's a lot of investment that they're putting in those drugs that make them, cost kind of what they do. But yeah, it's a lot of work.
I totally understand.
We were, yeah.
For someone with ALS, they're more concerned about the 13 years than the billions of dollars, because in a 13-year span, given 3 to 5 year diagnosis, that is generations of people with ALS who were diagnosed and passed away in that time.
You're absolutely right, and that's why this work has to, continue. And you're right that money's easier to get than time. We all have to be working on the at-bats, right? That's the preclinical kind of way you can look at it. We are creating the at-bats for clinical development, clinical trials. And we've got to keep that pipeline rich, both at ALS TDI and in biotechs and pharmaceutical companies across the globe, in order to, it has to be robust enough that the number of no's that we get still provide a number of yeses to keep, treating people over the course of time. Because not every person is going to respond to every drug, too. So it needs to be a lot of money in the preclinical space so that we know we have stuff coming up for people who are, diagnosed now and diagnosed in the future.
Which brings me to one last question here. We have a number of people with ALS from our area, ALS United Mid-Atlantic, who have participated in work through ALS TDI. We have helped fund some of that work as well. How can people with ALS and their families and caregivers help speed up that research? How can they be empowered and make an impact in these research studies?
Great question. So we need their help, and hopefully we can make it a pleasant experience for them. Through the ALS Research Collaborative, we have talked about some of the data that we collect from people living with ALS today. We've been collecting this data since 2014. And we will continue collecting it forever because it is really fueling research, like I said, at Mass General and across other places. So if people join this program, they're not just helping ALS TDI. They're helping research across the globe. And what it would entail is literally, maybe Tony, through this you can put the link up for ARC, as we call it, the ALS Research Collaborative, where people can register. And it's as simple as that. And then you will connect with someone in our ARC program, and we will determine what types of data you're willing to share. Maybe we'll be sending you those accelerometers that we talked about that you'll wear 1 week a month. Maybe you'll be calling that 800 number so that we can follow your voice. Maybe you'll be completing surveys so that we understand maybe what exposures or risk factors you had before you were diagnosed with ALS. We might send that phlebotomist to your home every couple of months to collect that blood so that we can look at NFL and define biomarkers of disease. So we understand that not everybody is going to want to participate in each aspect of the program. That's okay. If you want to do all of it, we'd love it. But the any data that anyone with ALS can give us is going to help ALS TDI and every research program on the planet, and we'll make it as easy as we can for you. And we share all of that data. We anonymize it. Nobody knows it's that particular person, but that data is shared with research programs so that they can advance theirs. So it's as easy as clicking register, and then somebody will connect with you, and they'll start to learn a little bit about your history and get you going on the program. It's really easy. We'd love that. And if you want to participate in clinical research, a way that we can help you make those decisions is we have a tool called the ALS Trial Navigator that allows people to put in what matters to them about participating in a clinical trial. Is it the placebo ratio? Is it proximity to their home? What is it? You can put in all of those factors, and it will spit out for you a list of clinical trials that might work for you, and then you can bring that into your clinician and discuss how to move forward. So that's an easy way to edit a list, because clinicaltrials.gov is confusing and intimidating. This is a much friendlier way to figure out how you can do it.
Well, I really appreciate it, and it really shows how people can be empowered no matter how they're connected with ALS, whether they are a person living with it, they're a family member, a caregiver, a donor, just someone who cares. There's so many ways to be empowered through research. If you're interested in learning more about what Carol and her team do at ALS TDI, you can go to ALS.net. It's a very simple website that's easy to spell and find. So ALS.net. Carol and her team have been involved with our Help and Hope events, and ALS United Mid-Atlantic is proud to support the research they're doing. Our website is alsmidatlantic.org, so you can go to our website and learn about our care programs, research, and advocacy in our region and how we're supporting the ALS cause. Carol, thank you so much for speaking today. I think your work is invaluable, and I'm proud that we can partner together for our ALS families.
Me too, Tony. Thank you so much for having me today. Thank you so much to ALS Mid-Atlantic, ALS United Mid-Atlantic, and all of your families for helping to support our work. We have an incubator that all of our nerds, and I use that term endearingly, are just thrilled to be able to speed up our work thanks to you guys and your support around that. We are very grateful. And thanks for being able to help this community understand that there are ways for them to engage that actually might be enjoyable and will help everybody. I really appreciate it.
So if you're listening, please subscribe to our podcast, our YouTube channel, give us a like and share, spread the message, because that awareness makes a difference. Thank you, Carol Hamilton, not Carrollton. Thank you, Carol Hamilton, and everyone at ALS TDI. Together, we're going to support our families.